11th episode of the BioLogic podcast. In this series, I've been talking about the cell, its structure, its organelles, how it produces energy, all that good stuff. Today, I'm going to be discussing how cells interact with each other, how they communicate and group together to form communities like tissues and, subsequently, entire organisms. In order for any organism to survive, its cells need to be connected to one another both physically and chemically. Physical connections allow groups of cells to maintain a continuous form without randomly falling apart like a clump of rocks. To pathogens, your skin is like a sealed barrier because of these connections. Chemical connections are not mechanical, but they're just as vital to the survival of the group of cells. Chemical signaling and regulation are required for groups of cells to manage their shared resources. Inefficient processing and distribution of nutrients and oxygen, ineffective waste management, or chronic hormonal misfiring will harm and eventually kill the tissue or organism. For example, when you belt a tree, you effectively kill it. By removing the outer ring of tissue, you starve the rest of the organism directly above that ring. The roots collect water and mineral nutrients, which are pumped up the vascular tissue of the tree. Belting the tree removes an outer strip of vascular tissue, basically destroying the nutrient input route for all the cells further up in the tree. Those cells then starve and die, and the whole tree dies shortly afterwards. Consider that the cellular membrane is the barrier separating life from non-life, the organized interior of the cell versus the chaotic and dangerous exterior. The cellular membrane is thus the interface between life and non-life, and it's upon this surface that physical connections between cells are built. This membrane interface is heavily studded with proteins, like pores, active transporters, glycoproteins, all sorts of stuff. Most cells also secrete a series of compounds onto their exterior, which forms a flexible shell or a coating. These compounds are called fiber composites, because they're, much like the structural carbohydrates I covered in episode 5, composed of fibers, and these fibers pile and link together to form sheets. In these fiber composites, the fibers are piled together into a sheet and covered in a surrounding material called a ground substance. This is very easy to understand in terms of concrete. Concrete is laid down as a heavy goop, which dries and solidifies. Steel rods are often crisscrossed within the volume of the concrete block, and the wet liquid concrete is poured around those rods. So the steel rods resist the tension of being pushed or pulled, while the concrete resists the compression of heavy objects. Together, these qualities make the concrete block a very sturdy and useful item. The fiber composites are very similar. The fibers give the cell a degree of resistance from tension forces, while the ground substance provides resistance to the pressure of adjacent cells. The types of fibers and ground substances used by cells have various strengths and qualities that make them better or worse at specific functions. Animal cells often secrete flexible compounds that can stretch or bend, while plant cells secrete compounds that can form stiff, rigid tissues like wood that can support tremendous pressure. In plant cells, this extracellular layer of fiber composites is a carbohydrate-rich structure called the cell wall. Young plant cells produce a primary cell wall, composed of crisscrossing bundles of cellulose fibers called microfibrils, and a gooey ground substance of sugars called pectins. Together, the microfibrils and pectin form a very strong sheet around the plant cell. The pressure of the water within the cell, called the turgor pressure, causes the membrane to get pushed up against the inside of the cell wall. In this way, the wall defines the shape of the plant cell. Some plant cells may, as they grow older, produce a secondary cell wall, which is secreted in a layer between the membrane and the primary cell wall. The compounds in the secondary cell wall vary enormously, not just between species, but between tissues within a single organism. The secondary cell wall in wood tissue is lined with an extremely strong and durable class of polymers called lignins. The secondary cell wall in leaf tissue is lined with a wax that greatly assists in reducing water loss. The secondary cell wall in stem tissue is stiffened with additional layers of cellulose, which provides greater structural support. If stem tissue was as sturdy as leaf tissue, for example, most plants wouldn't be able to hold themselves upright or move water from their roots to their leaves or flowers. Animal cells don't possess a cell wall, which is fortunate, because if we did, we would probably have a level of mobility on par with plants. Instead of a cell wall, animal cells secrete a much more flexible substance called the extracellular matrix. Where the walls of plant cells are carbohydrate-rich, the extracellular matrix of animal cells is protein-rich. The polymers in animal cells are protein fibers called collagen. The gooey ground substance that encases the collagen is composed of a swamp of proteoglycans, which are like nuggets of protein coated with numerous large sugar molecules. As in plants, the cells within animals also show variety in how their extracellular matrix is expressed. In tissues like bone and cartilage, there's a lot of extracellular matrix separating the cells. In skin cells, there's relatively little extracellular matrix separating the cells. Instead, they're connected directly through tight junctions and desmosomes. Cells that require a high degree of flexibility, like those in your lungs, use an elastic compound called elastin. Animal cells are connected to each other through the extracellular matrix. Cytoskeletal components on the inside of the cell form linkages to membrane proteins called integrins, which attach to a class of rope-like proteins called laminins. So the laminins interlink with one another to form a web or a net-like lattice across the exterior surface of the cell. This laminin net perforates and climbs through a layer of proteoglycans, ultimately connecting to larger collagen fibrils that run alongside multiple cells. Animal tissues are typically composed of millions of these structural collagen fibrils, collectively providing strength and great flexibility to the tissue. There are other mechanisms for cellular connection besides simply getting tied up into some big net. These connections are particularly important in epithelial cells, which are flat, thin cells, and they usually exist on the surfaces of organs or lining blood vessels to form smooth, sealed layers. Like the bilayer membrane is to the organelles within a cell, epithelial cells are to the organs within an organism. These cells form barriers within which specialized environments can be created. This is important because you wouldn't want your stomach acid, for example, leaking into your liver, or air from your lungs leaking into your heart. Both of those problems would cause you great pain and would inflict serious, most likely fatal damage. As you can see, it's important for epithelial cells to have tight connections, so they can form these tight seals. Two adjacent cells will typically have some sort of chemical adhesive holding them together. In plants, the space between the walls of two adjacent cells is called the Middle Lamella. Within the Middle Lamella, a gel-like layer of pectins holds the two cell walls together, much like peanut butter between two pieces of bread, or glue between two pieces of paper. In animals, this space is filled with a layer of goo, rich in proteoglycans and polysaccharides, which are similarly adhesive. Because animal cells don't have thick walls separating them, the cells can be grouped together much tighter and much closer to one another. This also allows for the formation of direct structural connections between adjacent cells. Remember the tight junctions and the desmosomes that I mentioned earlier? Those, as well as gap junctions, are some of the direct structural connections between animal cells. With that said, let's start with tight junctions. Tight junctions are regions where the cells are connected very close together, as the name suggests. Specific membrane-bound proteins on each cell connect with one another to form wavy rows of interlinking protein pairs. These rows are like stitches between two pieces of fabric, or like a row of staples connecting two pieces of paper. These tight junctions are indeed tight, often forming a waterproof or ion-proof seal between the cells. But they're weak and they can be easily broken. This is important for cellular function because it means tight junctions can be regulated. Loosening the tight junction encourages the movement of particles between cells, while tightening the tight junction encourages the movement of particles through the cells. Your blood vessels and bladder, for example, keep their tight junctions tight, otherwise they'd leak. Your intestinal cells, on the other hand, loosen their tight junctions during digestion to aid in the absorption of nutrients and then retighten afterward. By loosening, nutrients can move past the epithelial layer on the inside surface of your intestines to get absorbed by cells deeper in the tissue, and this improves the total efficiency and effectiveness of digestion by increasing the effective surface area. Desmosomes offer a significantly stronger connection between cells than that provided by tight junctions. Where tight junctions are similar to rows of staples holding two pieces of paper together, desmosomes are like groups of bolts holding steel plates together. Cells of a similar tissue type will connect together through their desmosomes, which are composed of three distinct parts. On the inside of each cell membrane sits a plate-like anchoring protein. Each anchoring protein is bound to a large number of smaller membrane proteins. These reach through the membrane up into the space outside the cell, where they connect with the other membrane proteins on the anchoring plate of an adjacent cell. These membrane proteins are called cadherins. They're responsible for the specificity of bonding between cells and the relatively high strength of the desmosome connection. So we have the anchoring proteins and the cadherins, which leaves the third part, the intermediate filaments. On the inside of the cell, loosely packed masses of intermediate filaments connect to the anchoring plate and run through the interior of the cell to connect to another anchoring plate somewhere else on the membrane. In this way, the intermediate filaments connect the desmosomes in a network that is simultaneously inter- and intracellular. When forces tug on one desmosome, the intermediate filaments spread out the force to the other desmosomes connecting to other cells. This provides a consistency to the organization of the cells within the tissue, as well as a physical consistency to the tissue itself. Gap junctions, the last type of direct cell-to-cell connection that I'm going to discuss, are not so much structural as much as they are communicative in purpose. The gap junction is a group of pores in each membrane, which align with one another to form tubes between cells. These tubes penetrate the membranes of both cells, allowing them to exchange small molecules and ions. In this way, adjacent cells within a tissue can communicate rapidly with one another by sending hormones or nucleotides or other small molecules through these little gap junction tubes. When the cells can communicate quickly in this fashion, they can coordinate their behavior. Consider your cardiac muscle cells, the cells in your heart that allow it to pump. If each cell contracted on its own, your heart wouldn't work. Your heart wouldn't contract to an irregular beat, it wouldn't contract at all. The individual contractions would make your heart look like a quivering, squirming mass, unable to forcefully pump blood. Without going into too much detail, your heart has a specialized group of cells called the sinoatrial node. This node produces an action potential which is sent to every cardiac cell, depolarizing their membranes and causing an influx of sodium ions. The gap junctions allow the sodium ions to be rapidly distributed across the cells of the heart, coordinating their contraction into a regular pulse. This coordinated contraction, enabled by the gap junctions, allows your heart to create regular, forceful pumps, which circulates your blood and keeps you alive. So gap junctions, tight junctions, and desmosomes are the close physical links between adjacent cells. This leads one to wonder, how do cells interact when they're far apart? Multicellular organisms like humans and elephants and pine trees, and even smaller organisms like mosquitoes, parasitic worms, and even multicellular microbes like tardigrades and rotifers, they're all huge when you look at them from the perspective of an individual cell. Cells are really tiny, so from their perspective in a multicellular organism, the distance from one end of the body to the other is absolutely tremendous. So how do distant cells communicate with one another? How do they interact as part of a single unified organism? The answer is signaling molecules. The concept of signaling molecules is pretty basic. When a cell or tissue needs to send a message to another cell or tissue, the sender will secrete a particular molecule. This molecule travels through the body, typically using the vasculature in plants and animals, although some smaller organisms just use diffusion to move the signal molecules through their tissues. This molecule is a signal molecule because it's sent from one place to another place, where it reacts with the cellular machinery to initiate a specific response. The message is received when the target tissue reacts to the signal molecule, stimulating a particular behavior, or depolarizing the membrane, or initiating a chemical cascade, or doing whatever it is that the activated cell or tissue does. If you're familiar with George Martin's book series, A Song of Ice and Fire, you might appreciate this analogy. The signaling molecule goes from one cell to another, causing a significant response. In Martin's book series, there is a place called the Citadel, and it's home to a group of scholarly monks. The seasons of this world are irregular and unpredictable, with winters and summers that sometimes last for years. When the summer ends and winter begins, those monks at the Citadel send a white raven, which is like a signaling molecule, to the major castles in the 7 kingdoms of Westeros. Once the castles receive this white raven, if they haven't noticed the weather yet, the nobility typically begins to make preparations for the potentially years-long winter. The Citadel is the source of the signaling molecule. The white raven is the signaling molecule. The castle is the receptor cell, and its rookery is the receptor. So when the signaling molecule binds to its receptor and initiates a response in the target cell, it's kind of similar to a bird keeper in the rookery taking the message to the castle nobility. The nobility begins preparations for winter. This is like the cell, whose response has begun, and the signaling is complete. Perhaps the most well-studied and well-understood group of signaling molecules are the hormones, small molecules that are secreted into the bloodstream, where they reach and interact with one or more target tissues to cause specific responses. Hormones are very powerful molecules. They need only be present in a very small concentration to cause a very potent response. Hormones exist in two large groups. They're either hydrophilic or they're hydrophobic. The hydrophobic hormones can interact with and pass through the hydrophobic phospholipid bilayer of the cellular membrane, binding to receptors inside the cell or to a specific organelle. Hydrophilic hormones can't pass through the cellular membrane, so instead they have to bind to receptors on the outside surface of the cell. The receptor is fundamentally important to cell signaling, as important or arguably more so than the hormone or signaling molecule itself. When a hormone is released into the bloodstream, it goes throughout the entire body. It comes into contact with everything. It goes everywhere the vasculature takes it to. However, the only tissues that actually bind and react with the hormone are those that have the right receptor for it. When multiple tissues have cells that express a receptor for the same hormone, each tissue will bind to the hormone whenever it's released. In this way, a single hormonal release can activate or stimulate responses in multiple tissues simultaneously. The cell's expression of receptors can change over time. Cells exposed to a very low level of a particular hormone may express more receptors in an attempt to capture any hormone molecule that it can. This is because the hormone comes by so rarely that the cell needs every chance it can to capture and bind to it. Conversely, cells exposed to a high level of a particular hormone begin to express fewer receptors. As the cell is frequently bathed in the hormone, it can afford to spend fewer resources on the receptors. Because it's swamped in the hormone, there's a certain reliability that the cell signal response will still be thoroughly stimulated, even if there are fewer receptors for the hormone to bind to in the first place. In cases like this, where a receptor experiences a high frequency of stimulation, it may find its ability to respond to the hormone gradually degrade over time. This happens in the brains of people who are addicted to and regularly take drugs like caffeine, cannabis, alcohol, nicotine, opiates, hallucinogens, and virtually everything else. Prolonged use, or rapid use in a short time period, creates a tolerance, and this means that the person must take more of the drug to achieve a similar effect. This is why some people need 3 cups of coffee to function properly, or 5 or 6 beers before they start feeling a buzz, or 2 joints before they start to feel alright. Receptors can also be blocked in the form of competitive inhibition. Some medicines and drugs work by competitively binding with these receptors, while others work by blocking the receptors entirely, preventing them from being activated in the first place. After a signaling molecule has been bound to a receptor, what does it mean to say that a response has been initiated in the cell? What is this response? And how does the receptor binding to the signaling molecule cause its initiation? First, we have to understand that the receptor is just a protein, and like any other protein, it'll often undergo some kind of conformational change when its substrate molecule binds to its active site. Hydrophobic hormones, or those that are lipid-soluble, will pass through the membrane and bind with a free-floating receptor bouncing around the cytoplasm of the cell. In the case of gonadal hormones like estradiol and testosterone, the hormone receptor complex is moved to the nucleus, where it gets involved in up or down regulating the expression of particular genes. In cases like this, the hormone is directly responsible for initiating the response. Hydrophilic hormones, in contrast, won't move through the membrane. These hormones have to bind to a receptor on the cell's outer surface, which reaches through or connects to proteins on the other side of the membrane, on the inside of the cell. Because the lipid-insoluble hormones have to use these membrane proteins as intermediaries for their signal, they operate indirectly. Signal transduction occurs when the information in the signal is passed through different mediums. In the first step, the information exists in a signaling molecule. After the signaling molecule binds, it causes the receptor's conformation to change. The information now exists in the altered conformation of the receptor molecule, and it's passed through to any protein that interacts with the receptor on the inside of the membrane. This may activate another protein, which activates another, and so on. The receptor mechanisms can be very complex, involving dozens of different proteins interacting to carry along the information. A protein at any step in this process may activate multiple proteins, which in turn activate more than the single protein ever could. This effect is called signal amplification, and it strengthens the cell's response to the hormone. Two major types of signal processing systems include G proteins and enzyme-linked receptors. Each type of system shows tremendous variety across cells in an organism and between species, but similar features exist between them. Each type of system shows tremendous variety across cells in an organism and between species, but similar features still exist. Some membrane receptors are connected to molecules called G proteins, which are studded in the membrane like anchors. The G proteins bind to GDP, a molecule similar to ATP, except it possesses the nucleotide guanine instead of adenosine. When GTP binds to a G protein, it causes a major conformational shift. This shift causes the G protein to disassociate from the receptor molecule, and then associate with a specific enzyme. The enzyme G protein GTP complex then catalyzes the synthesis of a new molecule called a second messenger. These second messenger molecules are small, able to diffuse through the cell rapidly. In this way, the enzyme G protein GTP complex can boost the signal from a signal hormone molecule into thousands of secondary messengers, which interact with various molecules in the cell to initiate a response. This is a textbook example of signal amplification. Secondary messengers like cyclic adenosine monophosphate activate particular protein kinases, molecules that catalyze the phosphorylation of other molecules. If you listen to episode 3 about proteins, I discuss how phosphorylation plays a major role in regulating cellular activity. Other secondary messengers like calcium ions bind to a receptor to form a complex, which then activates various proteins. The calcium ions bind to a receptor called calmodulin, and the calcium-calmodulin complex then binds to and activates or deactivates various proteins. It's important to understand that the same type of secondary messenger molecule may induce a different response in a different type of cell. Furthermore, a cell's response may require the actions of multiple different secondary messengers. Let's go back to the Song of Ice and Fire analogy that I used earlier. The white raven sent from the citadel is like a signaling molecule. The raven landing in the rookery is like the molecule binding to its receptor. The bird keeper is like the G protein, attached to the rookery receptor. He's stimulated with GTP when he receives the message from the white raven, and he goes to find a nobleman in the castle. So let's say our bird keeper G protein finds the lord, the ruler of the castle, and, basically analogous to our enzyme. The bird keeper gives the message to the lord, and in so doing becomes much like the enzyme G protein GDP complex. The lord of the castle then gives the news to all of his subordinates, his advisors, his servants, his officers and administrators, and much like the secondary messengers, these people then take the news to their specific departments and organizations and get to work preparing for the winter. Anyway, the other type of signal processing system involves receptors that are directly linked to enzymes. When the receptor binds to a hormone, the enzyme changes its conformation, or in the case of the receptor-linked tyrosine kinases, or RTKs, it causes it to form a dimer. The altered receptor-enzyme complex is then able to be phosphorylated, or it can phosphorylate itself, and this attracts a G protein. In this way, the enzyme-linked receptor process works in the reverse direction of the G protein process. The G protein is activated by the receptor-enzyme complex, which causes the phosphorylation of a different protein, a protein kinase. This protein kinase then causes the phosphorylation of another protein, which does the same to another protein, and so on and so forth in a phosphorylation cascade. In many cases, one kinase can activate multiple kinases, which causes signal amplification. There's one more step involved in cell signaling: shutting the signal off. It's all well and good to send a signal when you need to, like a developmental hormone released at a specific time in the fetus, or the surge in sex hormones during puberty, but it's equally important to shut off this signal when necessary. Failure to shut off a signal during fetal development can lead to deformities and health complications, if not outright death. Interfering with one or more proteins involved in the processing and amplification stages can usually downregulate or shut off a signal. For example, phosphorylation cascades can be extinguished with molecules called phosphatases, which dephosphorylate and thus deactivate many different proteins involved in the cascade. Second messengers can be metabolized or cleaned up, like when the smooth endoplasmic reticulum cleans up and stores calcium ions. Breaking the GTP on a G protein down into GDP will deactivate it and prevent it from binding to an enzyme to create more secondary messengers until it's reactivated. In this way, a sufficient effort to dephosphorylate proteins in a phosphorylation cascade, or to clean out secondary messengers, or to deactivate G proteins will lead to a cessation of the signal. Perhaps the most famous example of a signal failing to be stopped is cancer. Tumors are caused by cancerous cells that cannot turn off the signal to keep dividing. The cancerous cell will divide and divide and divide, consuming resources and growing into a larger mass of cancer cells. Benign tumors are those that are encapsulated, or their growth has slowed or stopped. They don't spread. Malignant tumors, on the other hand, aren't encapsulated. They replicate aggressively, and they spread. Chunks of cancerous cells can break off of malignant tumors and float through your bloodstream, lodging in other areas where they reproduce and form new tumors. Benign tumors often look like lumps or growths, which can usually be removed with surgery. Malignant cancer, on the other hand, can form tumors almost anywhere in the body, requiring chemotherapy to treat. Surgery is far too brutal and invasive of a method to be used to remove malignant tumors, as there can be dozens of them scattered all over the body. Being able to stop a signal or downregulate its amplification are important facets of response regulation. You see, cells respond to various signals almost constantly. They respond to many different kinds of signals and hormones at the same time, processing them and performing the necessary responses. In many cases, the G proteins, enzymes, and secondary messengers in one signaling pathway can interact with those in a different signaling pathway by up or downregulating their expression. A hormone can bind to a receptor and activate an enzyme in a particular signaling pathway. This enzyme could be used to continue the pathway, or it might be able to inhibit some protein in a different pathway. This would reduce the signal strength of the second pathway, even though the initiating molecule is still present. Alternatively, the secondary messenger in one pathway may stimulate some proteins in a different pathway, encouraging a stronger signal response in that different pathway. As each signaling pathway involves numerous steps, this gives numerous opportunities for various pathways to regulate one another. Because cells are always being sent some kind of signals, there is always some kind of interaction between the active signaling pathways. Each pathway performs its particular functions, including using its own products to regulate the performance of the other pathways. This chemical crosstalk between signaling pathways is responsible for sustaining the health of the cell in a dynamic and carefully balanced way. Even small fluctuations in the concentrations of signaling molecules can have enormous downstream effects on the cell. That should cover most of the very basics of cell interactions. We started with cell walls and extracellular matrix, then junctions and desmosomes, and ended with hormones and signaling pathways. Before I end the episode, I want to briefly discuss a really fascinating thing called quorum sensing. All of the interactions I described in this episode exist between multicellular organisms, but colonies of single-celled organisms can interact through quorum sensing. Small molecules are released into the environment, and they diffuse to reach any proximal individual cells. In this way, colonies of single-celled organisms, particularly densely packed colonies, can communicate their situation and coordinate their activity in response. Quorum sensing allows colonies of single-celled organisms to behave in a manner eerily similar to the tissues of a multicellular organism. This even allows cells of various prokaryotic or protist species to engage in complex group behaviors, like moving across a surface or arranging into a fruiting body. Alright, well, I think now that's pretty much everything. This was a fun episode. I hope you enjoyed listening to it as much as I enjoyed doing the research, writing, and notes, and making the recording. I certainly hope you learned something cool, something that you can talk about at the dinner table or with your roommate, or to your dog if you have no one else. Thanks for listening. If you enjoyed this episode, hit the like button and subscribe. Share the episode with a teacher, a friend, or a classmate. If you want to support the show, follow us on social media and consider buying something from the official store. Follow us on Twitter. Our username is @BioLogicPodcast. To buy something from the store, go to redbubble.com and search for BioLogic. Or go directly to the store at redbubble.com/people/biologic/shop. Become an official patron of the show. Sign up at patreon.com/biologicpodcast. Your support really helps the show, so if you like it, send us some love. And as always, thanks for listening.